So this video is going to be the first of two parts explaining genetics. The outline of this presentation is going to be explaining what genes are and how they make proteins, going into the structure of genes and the types of mutations that can occur in genes, using analogies with sentences, which are a little easier to understand, and then understanding the different types of mutations and how they're specified in a report of a genetic test. So DNA consists of, you've probably heard of the double helix. It has basically two strands that are bound together, and each strand goes in the opposite direction. And there are a series of 4 different bases on the DNA, abbreviated A, C, G, and T. And an A on one strand is always paired with a T on the other, and a C is always paired with a G. So a gene is a particular region of DNA which contains the instructions for making a specific protein. You can think of it as a gene is a recipe for making that protein. Now, not all of our DNA is actually part of a gene. Only a small proportion is. So we have about 26,000 genes in the human genome that our bodies are able to make the proteins corresponding to those 26,000 genes. Okay, so what is a protein? Okay, so a protein is a chain of amino acids. And you can, think of, protein in your diet. That's a protein from some plant or animal. And when you digest it, you break up the protein into its component amino acids. Then your body uses those amino acids to assemble your own proteins, following the instructions in your genes. So you can eat it. It's not a matter of how much protein you eat. If you don't have the proper instructions for making a certain protein, your body won't be able to make it. Okay, now there are 21 different amino acids that are used by our bodies to make our proteins. And conveniently, that's slightly less than the number of letters in the alphabet. So there's this abbreviation, which you can memorize if you want to pause the video, but you don't have to. It's sort of semi-intuitive. Each amino acid is abbreviated by a different letter. Now, in addition to all the amino acids, there's also, something called a stop, which means that the DNA tells the body, okay, this is the end of the protein. We're done making this protein. So a protein is made in 3 steps. There's what's called the expression of the gene. Now, all of the cells in our bodies contain all of our genes, but not all cells make all proteins. So if a cell is going to make a certain protein, the first thing it does is to actually copy the DNA for that gene into a different form called RNA, and that's actually what is used to make the protein. Then it turns out that the way that the DNA is organized in our genes, the RNA needs to be spliced together so you have something that can be turned into a protein. And then that RNA sequence is translated into an amino acid sequence. So the way that works is that every 3 bases of DNA, which is called a codon, specify a particular amino acid or a stop codon. And this is the genetic code that's been worked out many years ago. Okay, so for a particular protein, I'll use the dysferlin protein as an example. The gene starts with what's called a start codon, and that is ATG, which specifies the amino acid M. And it turns out every gene starts with those 3 DNA letters specifying an M. Okay, then each group of 3 DNA letters in a codon specifies another amino acid, M, L, R, V, etc. 2,000 amino acids later, you come to a stop codon, which marks the end of the protein. Okay, so for differences in DNA between different people, okay, by and large, people all have the same genes. So colloquially we say, oh, I have the gene for this, or that causes me to be a certain way. Really what that is we all have the same genes, but we have variants in the genes. And a variant just means any change in a particular person's DNA from a quote-unquote standard sequence. We all have DNA variants. Now, for most of the time, the variant doesn't really cause a problem. It just makes us different than someone else. And in that case, it's called a polymorphism. If it's something which can cause a protein to not work correctly, that might cause a disease, it's called a mutation. And sometimes in practice, we see that someone has a change in their DNA, but we don't always know what it means, and that's called a variant of uncertain significance, which is abbreviated either VOUS or VUS. So to illustrate different changes in DNA, we're going to use a sentence analogy, which is a little bit easier to follow than a bunch of ACs, Gs, and Ts. And so our sentence is going to consist of 3-letter words, because every 3 bases of DNA makes a codon which specifies a particular amino acid in our protein sequence. So our sentence is going to be, the cat saw the rat, but not the bat. And then there's the word out, which actually isn't really part of the sentence, but it's a code word meaning that this is the end of the sentence. And so then we're going to, go through different changes in this sentence and show, how they're captured in DNA. Okay, so the first type of change we're going to illustrate is a polymorphism. So an example of this would be if you change the spelling of cat and spell it with a K rather than a C. Okay, it's a little bit different, but the meaning of the sentence is still clear. Everyone's going to interpret it correctly. And so, our protein here, which is illustrated by a little cloud, is still basically looks the same as it did before. Okay, so this is the end of part one. So in part two, we'll go through other types of mutations in DNA that do impact the protein that's ultimately produced. So thank you for watching.